Hey everyone, welcome back to The Explainer. So let's just jump right into it. If you're studying computer science, you've probably already played around with AI tools, right? Having them write code, debug scripts, maybe even spin up a basic web app for you. But what happens when we take that exact same AI and assign it a totally different role? What happens when we treat our most advanced AI models as basically junior developers in the incredibly complex, super messy world of biology? Well, that's exactly what we're going to evaluate today.
I mean, it's kind of the ultimate question right now. If our AI interns can write perfectly clean Python or C++, can they eventually help us cure diseases? Can they handle the messy, ambiguous, totally real-world data of biology? Because, let's be honest, the jump from generating a simple sorting algorithm to actually uncovering the genetic markers of a disease, that is massive. So we really want to know if today's frontier models are actually ready to bridge that gap.
To understand this, we first need to talk about bioinformatics. And if you're a freshman CS student, the easiest way to think about bioinformatics is simply this. It's data science for biology. It's a whole domain where researchers use computational tools, statistical models, and massive data sets to uncover new biological knowledge. And the cool part? This often happens completely independently from traditional wet lab experiments. You aren't standing in a lab coat holding a pipette. You're sitting at a keyboard, parsing terabytes of genomic data.
But we absolutely have to make a crucial distinction here. Standard software engineering is very, very different from biological data analysis. Think about it. In standard coding, a syntax error throws a clear, undeniable flag, right? The code either compiles or it doesn't. A test either passes or it fails. Biological data analysis, on the other hand, is literally like dealing with the universe's ultimate spaghetti code. It is wildly open-ended, highly ambiguous, and just filled with noise.
And this brilliantly illustrates the reality of the field, which was highlighted recently by Anthropic's BioMysteryBench research. They pointed out that biological data is incredibly human difficult to debug. Why? Well, because in biology, there are just so many different right ways to do something. Individual research decisions are incredibly subjective. Like a researcher's subjective choice on how to filter noisy datasets can lead to entirely different conclusions. Now, to be fair, Claude, Anthropic's AI, has shown some real promise here. It's actually solved biological mysteries that completely stumped panels of human experts by tapping into its huge knowledge base and cross-checking multiple analytical methods. But measuring that capability systematically, that is a huge challenge. Which brings us to Section 1, BioMysteryBench, the Integration Test Suite. So if we want to see how good AI really is at bioinformatics, we need a super rigorous testing environment. Enter BioMysteryBench. This is a massive suite of complex integration tests specifically designed for AI agents. Expert bioinformaticians literally built out 53 real-world analytical scenarios just to serve as test cases. Human analysts assembled code notebooks, provided the input data, and established ground truth results, which ultimately generated nearly 300 open answer questions that require true, deep analytical reasoning to solve. And the biological code base that the AI is being tested against is incredibly diverse. We are talking about a scope that ranges all the way from genomics and transcriptomics right down to imaging and single-cell analysis. It is a wildly comprehensive exam covering a massive scope of computational biology. Moving on to Section 2, how the AI operates. Recent demonstrations on YouTube have actually shown AI, like Claude Code, acting directly inside an IDE like VS Code. It acts like a pair programming intern, autonomously planning, writing, debugging, and updating a complete RNA data analysis project in just 1 hour. It's wild. And BioMysteryBench uses a brilliantly similar setup to test agents in a standardized way. The AI is dropped into a Dockerized Jupyter environment, preloaded with popular bioinformatics packages in Python, R, and Bash, alongside an empty notebook and a folder of data. To navigate this whole environment, our AI intern is given a specific toolkit. Basically, it has 3 main commands at its disposal. First, there's edit_cells, which allows the agent to write, modify, and actually execute code right within the notebook. Second is list_directory, which lets it inspect the workspace and understand the structure of the dataset. And finally, submit_answer, which it uses when it has totally finalized its analysis and is, ready to be graded. So, the absolutely crucial point here is this. There is no human holding its hand.
None. The AI must independently explore the data. It has to make its own assumptions about how to clean that data, plan its own analysis, write the code, debug the totally inevitable errors, and execute long analytical trajectories, exactly like a real human researcher would have to do.
Which leads us to Section 3, failing the integration test.
Okay, so the setup sounds amazing, right? A tireless AI intern crunching data perfectly all day long. But how did today's absolute top frontier models actually perform when put through this intense integration test? 17%. That is the surprising climax of this research. The most advanced models available today achieved a maximum of only 17% accuracy on these open-ended, real-world bioinformatics tasks. To put that in CS terms for you, if this was your final project, your AI partner basically just bombed the integration test. When looking at the data in the open answer regime, which really mimics real science because, there are no multiple choice hints in reality, both models struggled tremendously. If you look here, notice how the performance just hovers right around that solid gray line. That line represents random baseline recall. So without the crutch of multiple choice options, autonomous scientific discovery is clearly still a massive challenge for these models. Now, comparing our two specific AI interns, Anthropic's Claude 3.5 Sonnet definitely performed better, hitting that 17% mark. OpenAI's GPT-4o, on the other hand, only managed a 9% accuracy in the open answer setting. So while Claude performed nearly twice as well, the overarching takeaway is that both models still essentially failed at the integration test. The noisy, highly ambiguous reality of biological data just proved way too complex. And there is a rather humorous quirk in the data regarding how the AI handled all this complexity. The researchers actually tested a multiple-choice version of the exam, and when they gave the AI a refusal option, basically an "I have insufficient information" choice, the models actually performed worse than random guessing. When the data got too confusing or too ambiguous, the AI interns tended to just throw their hands up, opt out, and refuse to answer entirely. Pretty relatable, honestly. There's also another fascinating bug in the system here. The AI actually really struggled to interpret visual plots in the notebooks. So much so that when researchers forced the AI not to generate image plots, completely restricting it to text and data tables, its performance actually trended higher. The vision models literally performed better when they were forced to ignore visuals entirely and stick purely to code and text outputs. How crazy is that? Let's wrap things up with Section 4, debugging the future. So, does all this mean the dream of the autonomous AI bioinformatician is just dead on arrival? Not at all. The researchers who built BioMysteryBench do not view this 17% accuracy as a permanent failure. In their conclusion, they state that it highlights a fundamental shortcoming, which will serve as a super valuable resource to guide future development. Honestly, think of this benchmark as a roadmap. It's the ultimate unit test, so developers know exactly what bugs need to be fixed for the next big AI upgrade. Which leaves us with this final thought. What is it going to take to merge the strict syntax-driven logic of AI with the noisy, beautiful mystery of biology? The gap is clearly wide today, but as we all know, the first step to optimizing any code is writing the tests to expose the bugs. For any computer science student watching right now, this right here is the frontier. The tools are there, the IDEs are ready, and the biological mysteries are just waiting to be solved. Thank you so much for joining me for this explainer. I hope it sparked some curiosity, and I will catch you in the next one.